Welcome to AP Biology. This video is a tour through the cellular organelles. We're going to talk about many different organelles within the cell, but all organelles have the same basic function. The major function of a cellular organelle is to compartmentalize chemical reactions that occur within the cell. So they allow chemical reactions to occur in very specific locations. They do this by isolating the specific enzymes responsible for those chemical reactions. We'll see that many of these organelles are highly folded. By having highly folded organelles, it's going to increase the surface area within the cell, allowing more to happen in a smaller space. The first organelle is going to be the nucleus. The major job of the nucleus is to hold the DNA. The DNA contains the entire genetic blueprints on how to create that organism. Inside the nucleus, the DNA is wrapped around proteins to make a material called chromatin. Chromatin is the material that our chromosomes are made up of. Each human cell inside the nucleus will contain 46 chromosomes that hold our DNA, our entire genetic code. The nucleus contains a nuclear envelope. That's the membrane covering the nucleus. The nuclear envelope is made up of phospholipids, just like the cell membrane. The nuclear envelope contains nuclear pores, and these nuclear pores are going to allow things like messenger RNA to leave the nucleus. The shape of the nuclear envelope is maintained by the nuclear lamina. The nuclear lamina contains pieces of cytoskeleton, which you see right here, which is going to maintain the structure of the nucleus. Also contained within the nucleus is a dark structure called the nucleolus, which you see right here, which is responsible for the production of ribosomes. Overall, the most important function of the nucleus is that it holds all of the cell's DNA. The cell's DNA contain all the genes necessary for the production of that organism. The DNA contains the blueprints for the creation of all the proteins within the cell. The proteins are actually made at organelles called ribosomes. Ribosomes are the organelles responsible for carrying out the process of protein synthesis. There are two types of ribosomes. You have free ribosomes and you have bound ribosomes. Free ribosomes will be found in the cytosol, where bound ribosomes are attached to the rough endoplasmic reticulum. Generally speaking, both of these ribosomes do the same thing. Free ribosomes tend to produce proteins that are used within the cell. Bound ribosomes tend to produce proteins that are going to be exported out of the cell or secreted from the cell. Now we have the endomembrane system. The endomembrane system is going to be highly involved with the process of protein synthesis. It's going to be made up of many organelles that are all made up of pieces of membrane. So they're all made up of phospholipid bilayer, just like the cell membrane is. They include the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, vacuoles, and the plasma membrane itself. All of these components are continuous, meaning they can communicate with each other and send stuff to and from each other using structures called vesicles. A vesicle is simply just a sac, which is also made up of membrane. The first members of the endomembrane system we're going to discuss are the endoplasmic reticulum. There's two types of ER. You have the smooth ER and you have the rough ER, and they have different functions. I would say the major function of the smooth ER is in detoxification of drugs and poisons. The smooth ER is also involved with the production of lipids like oils, phospholipids, and steroid hormones. The smooth ER is involved in carbohydrate metabolism, the breakdown of large carbohydrates, and it's also used to store calcium ions. We'll be seeing later on in the course what function calcium performs within the cell. The rough ER is called the rough ER because it contains bound ribosomes, giving it this studded appearance. The rough ER really functions in the process of protein synthesis. After proteins have been made on the bound ribosomes, those proteins will enter the rough endoplasmic reticulum, where they will be modified. The rough ER is going to secrete, or it's going to release both proteins and also glycoproteins. Glycoproteins are proteins that have carbohydrates bonded to them. When the rough ER secretes or releases a protein, it's going to release it using a transport vesicle. That transport vesicle is going to move the protein from the rough ER over to our next organelle, the Golgi apparatus. Besides functioning in protein synthesis, the rough ER is also going to be responsible for the production of phospholipids. It's a membrane factory for the cell. Here we get to the Golgi apparatus. I think of the Golgi as being the UPS of the cell. It is going to finish, sort, and ship proteins to their final destination, either somewhere inside of the cell or export the protein out of the cell. Some common proteins that get exported out of cells are proteins that function as hormones. You have proteins that function as local regulators. You have proteins that function as neurotransmitters. We'll talk about them later on in the course. The Golgi apparatus has two faces, or two poles. One is called the cis face. That's the receiving face. That's where the transport vesicle is going to come into from the rough ER. And it has the trans face, which is going to be the shipping face, the exporting face. Each of the individual pieces of the Golgi apparatus is called the cisternae, and again they're just simply made up of phospholipids, of membrane. Lysosomes are going to be sacs, like a vesicle, but it's a sac that contains digestive enzymes. Enzymes that perform the process of hydrolysis and break things down. Lysosomes are used for many functions. One function is intracellular digestion, breaking down things within the cell, things that are no longer needed or no longer functional. Lysosomes also participate in a process called autophagy. Autophagy is the recycling of cell material. If we look here on the right, here we see the process of autophagy. You have a lysosome containing the digestive enzymes, and here you see unused organelles being shipped to the lysosome. You see a peroxisome, you see a mitochondria, and you can see here that the lysosome is going to fuse with the vesicle, and the digestive enzymes are going to break down the mitochondria and the peroxisome, and the cell will be able to reuse its components. It's going to be able to recycle the cell material. Lysosomes are also going to be involved in a process called apoptosis. We're going to see apoptosis throughout the course. Apoptosis is programmed cell death. Some cells are actually programmed to destroy themselves. This happens during different stages of embryonic development. It's how we lose the webbing between our fingers. This can also be triggered by mistake. Mistake during the cell cycle that can lead to something like cancer forming. Instead of that happening, the cell can detect the problem and destroy itself. Apoptosis is an extremely important process that can take place. And we'll see it over and over and over and over again. Some cells actually have the ability to engulf other cells. We call this process phagocytosis. An example of a type of cell that does this is the macrophage, a large white blood cell that's part of the immune system. When a macrophage engulfs another cell through phagocytosis, the cell membrane is going to indent and wrap around that object, and it's going to form a vesicle that you see right here, a sac of membrane, that we call the food vacuole. The macrophage could use this to engulf something like a bacteria. That food vacuole will then travel to the lysosome, it will fuse with the lysosome, and the digestive enzymes inside the lysosome will destroy that bacteria. A vacuole is very similar to a vesicle, but it's really used for storing something. Food vacuoles we just talked about, they're created through phagocytosis. Some protists have contractile vacuoles. An example of a protist that would have this is a paramecium. They use their contractile vacuole to pump excess water out of their cells. They use this to maintain proper water balance. Plant cells have a large central vacuole, and this is used primarily to store water. It's also used to hold other organic compounds. Here you see a plant cell, and you see just how large that central vacuole is. It can cause a plant to become turgid, swollen if it's filled up with water, or flaccid and wilt if it is empty of water. Another organelle as part of the endomembrane system is the peroxisome. The peroxisome contains two enzymes, one called oxidase and one called catalase. The major function of the peroxisome is to perform a particular oxidation reaction called beta oxidation. With beta oxidation, fatty acids enter the peroxisome, and using oxygen, the enzyme oxidase breaks down that fatty acid into two carbon molecules that could then enter cell respiration and be used to manufacture ATP. As a waste product of this oxidation reaction, hydrogen peroxide is formed, which can be toxic to the cell. The peroxisome also contains the enzyme catalase, and the job of catalase is to take this toxic hydrogen peroxide and to break it down into water and oxygen gas. Here we see how the endomembrane system is involved with the process of protein synthesis. We begin at the top here with the nucleus. The nucleus contains all the DNA, the blueprints on how to create all of the cell's proteins. Protein synthesis is actually going to take place at these tiny little dots that we call the ribosomes. Once these bound ribosomes manufacture the protein is going to enter the rough endoplasmic reticulum, which is going to modify that protein. After that protein's been modified, you can see here a transport vesicle is going to form, and that transport vesicle is going to ship that protein to the Golgi apparatus. It's going to be received at the cis phase. It's going to be further modified. It's going to be packaged, and it's going to be shipped out using the trans phase. That Golgi apparatus can either ship that protein somewhere within the cell that it's needed, or it can secrete that protein from the cell, just like you see right here. Two extremely important organelles within the cell are the mitochondria and the chloroplasts. They are so important because they are energy transformers. Mitochondria is the organelle in which organic molecules are broken down to regenerate ATP. Organic molecules are not actually a usable energy source within the cell. Cells can only use ATP to power their chemical reactions. The mitochondria that you see right here is the organelle used to produce all the ATP within the cell with the use or in the presence of oxygen. The chloroplast is found in plant cells. It's also found in algae, and it is going to convert solar energy into chemical energy, the usable energy within the plant cell. Where did mitochondria and chloroplasts first come from? Scientists believe that mitochondria and chloroplasts were once their own separate prokaryotic organisms. They believe they were once their own bacteria, and these bacteria were engulfed by a larger cell, and they formed a permanent relationship. There's lots of evidence for this idea. Number one, mitochondria and chloroplast both contain their own circular DNA. Remember that only bacteria have circular DNA. Mitochondria and chloroplasts have their own ribosomes. Mitochondria and chloroplasts reproduce independently of the cell that they are found within. So they're like their own separate organism living inside the larger eukaryotic cell. Both chloroplasts and mitochondria have a similar structure. They both contain a double membrane. The theory that describes how these two cells formed a close and permanent relationship, the theory that describes the origin of mitochondria and chloroplast, is called the endosymbiotic theory. Here we see a picture of what happened. We begin with the ancestor of all eukaryotic cells. It's very large, but it would not be able to become very complex because it did not have a rich source of ATP. It didn't have the fuel that it needed to run all of its chemical reactions. This large cell engulfed a smaller cell through phagocytosis. This small bacteria that was engulfed was an extremely efficient energy transformer. It was able to take in food and convert that food into lots of ATP in the presence of oxygen. Both of these cells benefited from each other. The larger cell gained a steady supply of ATP. The smaller cell gained protection, and it also gained a steady supply of food. This could also have happened with the chloroplast. These relationships were so beneficial that they became permanent over time. And now these mitochondria and chloroplasts are now considered permanent additions of eukaryotic cells. This completes the video.